Now we have talked three types of main cancer genes: the proto-oncogene and oncogene, the tumor suppressor gene, the cellular functions and how they activate it or inactivate it. Then we are talking about the third types of third category of cancer genes, which is the DNA repair gene. And here's an example of BRCA1 and 2 and its mutation, how the mutation leads or increase the risk of breast or ovarian cancer. And another example of DNA repair gene is involved in human non-polyposis colon cancer. And this is the types of rare types of colorectal cancer is like 2 to 3%, and it's also autosomal dominant and it's with high penetrance. And there is a typical age of this type of cancer onset is like to 40 to 50 years, and it's caused by DNA repair genes called misrepair genes in MSH2, MLH1, MSH6, and so. And also the tumor formation requires mutation at the second allele. That's why a lot of people just classify the third category of genes, like DNA repair genes, into tumor suppressor genes. And blood tests are available for all these four genes. As you can see from this figure, the risk with cancer, the other risk with cancer for patients in the HNPCC, for example, the colorectal cancer and the endometrial cancer will increase dramatically with the increase of age. So this is the three types of cancer genes. And as you have learned, that the cancer is a genetic disease arises from the malfunction of genes, and majorly involved in these three categories of genes that I have talked about. So then, how a tumor is initiated and progress by driven by these tumor genes. So now it's well accepted that tumor initiates from a single founding cell. As shown here, this cell by accident it will acquire a mutation in the epithelium, and this cell will proliferate, and then it will acquire another mutation, and this mutation will foster the cell division and proliferation. And in this repeated rounds of mutation and proliferation, which then the cell will acquire more mutations and gain more proliferation advantage. In the end form the malignant tumor cells and dominate the tumor mass. This is the process of tumor clonal evolution. It essentially starts with a single called founding cells, and by repeated rounds of mutation and proliferation, and then one type of malignant tumor cells will dominate the tumor mass. And as you can see here, this is a simple plot with the colon cancer incidence as a function of age. You can see that with the increasing of age, the colon cancer incidence that is increased very sharply. That means this cancer or colon cancer takes some time to happen. It also suggests that it requires accumulation of several mutations in these cancer genes to acquire cancer. So the trend for other major types of cancer is essentially the same. All the major types of cancer, the incidence is just increased with the increasing age. You can imagine that in case one can live long enough, in the end you will you will definitely get cancer. And as shown here, 3 examples shows how these tumor genes or cancer genes involved in the progression of cancer. The first example is colon cancer. So here, this is the well-structured normal colonic mucosa, and then you loss of the APC gene, which is a tumor suppressor gene, and you will get the benign, the adenoma, and during the progression, you will have to activate another oncogene, that is the RAS oncogene, and you will get more cell proliferation and still confined to the territory. And then you will continue to lose the tumor suppressor gene P53, and also other cancer critical genes. In the end, you will get the malignant invasive carcinoma. And for this process, from the normal situation to the final invasive carcinoma, it normally takes like 10 to 20 years to really develop a colon cancer. But this is not include that you have an inherited mutation from your family. And the second example is the astrocytoma. This is a very common and rapidly developing brain tumor, and it also, you can see here, from normal to malignancy, it requires a couple of changes of tumor suppressor and as well as some loss of function of tumor suppressor gene and activation of some oncogenes. And in the last example, I would like to emphasize that the tobacco smoking, cigarette smoking, and the development of lung cancer. And here shows the healthy lung, and the basal cells and the basement, and also the collective tissues. So after 1 year of smoking, heavy smoking, the basal cells start to proliferate, and then after like 5 years of heavy smoking, you see that this basal layer cells just change the type. They start to proliferate. And then in 8 years, you will see that more cancer cells with an abnormal nuclei. And then these cancer cells will finally invade the basal layer and the collective tissue. This is forming invasive malignant tumor, and the whole process normally takes like 20 years. So now it's quite clear that the heavy smoking, the tobacco in the cigarette smoking, has a clear relationship with the lung cancer. And remember that lung cancer now is the leading cause of cancer deaths, because there is no effective treatment for lung cancer so far. Okay, just have a summary of the part 2 about what causes cancer. I have showed you that cancer is genetic diseases that arise from the malfunction of genes, and both these external causes like chemical and UV radiation or infection have demonstrated genetic changes linked with cancer, and the intrinsic causes is clearly genetic. And then I introduce you these 3 main types of cancer genes, including oncogenes, tumor suppressor genes, and DNA repair genes. Sometimes a lot of people just classify the third category into the second category because they have a similar way of inactivation. Both of them need to be inactivated to really promote tumor. And in this I give you the definition of the 3 categories of cancer genes and the cellular functions, and then the mechanism in case of proto-oncogenes, mechanism of activation, and in case of tumor suppressor gene and DNA repair genes, is the mechanism of inactivation. And I give you some very famous and representative examples of how these three categories of genes or not need to the initiation or progression of cancer. And in the end, I'll discuss with you how these cancer genes cooperate, to initiate and progress to finally lead to cancer. Okay, with this knowledge, I will come to the third part of my talk. So we have now known that cancer is a very complex disease and have a relatively clear genetic reasons, and we know what cancer genes are involved in cancer development and cancer disease. Then the question is that how can we use this knowledge to prevent and diagnose or even treat cancer? So for cancer with known reasons, we have developed very effective way to prevent and diagnose cancer. For example, for the liver cancer, a lot of quite a proportion of liver cancer is caused by the hepatitis B virus infection. Now we have a vaccine to this virus, and the liver cancer actually indeed dropped quite a lot. And also we now have more healthy food, try to avoid this mold in the food. And also we know that the smoking has a clear relationship with the development of lung cancer, so we advocate to be away from smoking. And for cervical cancer, now we have the pap test that you can, by this cytology screening, that you can view the cells from the cervical, and also have the HPV test. And these tests are very efficient to prevent this cervical cancer. And for the colon cancer, we have now very mature colonoscopy. And for the cancers with clear genetic reasons, particularly for some heritable or familial cancer syndromes, we have developed the genetic screening. For example, we have the APC screening in the familial adenomatous polyposis. This is a rare type of colon cancer. And then we have the BRCA1 and 2 in breast and ovary cancer. And also we have sets of cancer predisposing genes in clinic for screening. Now with this, I would like to point out and emphasize that in terms of cancer, the prevention and early stage detection is far more effective than treatment. Actually, till now, there are not many treatments or drugs that can really cure cancer, but the prevention and early stage detection can really prevent it. So here I showed you how this pap test is applied in the clinic for cervical cancer. And for this HPV, you have several subtypes. We can actually detect it by simple DNA hybridization for the HPV test. But in the earlier stages, for example, for women, we start from the age of 20, we will have this pap test, maybe every 3 years or every 5 years. And the pap test is very easy cytology way to detect these precancerous cells. So as shown here, this is the normal cells that you scraped from the cervical. And then in the B, you can see some precancerous cells, and C, this is the invasive cancer cells. So we need to monitor this and to prevent this cancer development from here. And also we can decrease the risk factors like smoking or having many children, but this is not really needed. And also the HIV infection or these risk factors. And for the genetic test of breast cancer, you may well know that this is so-called Angelina effect. Maybe you are very familiar with this pretty and very, in my opinion, very brave lady. And last year, she actually took the genetic test for a set of cancer predisposing genes. And this set of genes that cost her like $4,000. And in the end, she found that she has a BRCA1 gene mutation, and also she had a family history of these cancers. So she and I think her doctor made a decision that she agreed to take her breast surgery. And I think this is from the medical point of view, this is a necessary action that need to be taken. And after this Hollywood star has taken this genetic test, and more and more women in US and UK or other developed countries have used the genetic test. Okay, let's see the cancer death rate for different types of cancer. As you can see here, the stomach cancer, by avoiding this contaminated food, the stomach cancer has decreased very much, or by avoiding this bacteria infection. Also for the liver cancer is the case. And the breast cancer. Okay, where's the breast cancer? No breast cancer here? No. Okay. And you can see, okay, breast cancer is for women. Sorry. Okay. It start to decrease. Also start to decrease. But the problem is that you can see that this is non-cancer. Non-cancer increase dramatically and then start to decrease a little bit. It's now the non-cancer is the leading cause of death for both male and female. And all this decrease is clearly maybe has a relationship with the effective prevention or effective earlier stage diagnosis. So now, thanks to the revolution of DNA sequencing technology, these are the first generation and next generation or the third generation sequencing technologies. In the conventional way, you can just have very limited sequencing capacity, but now you can sequence the human genome with a very relatively acceptable cost and very short time. So, for example, this Illumina machine can sequence the human genome like within 1 week, and cover the human genome like hundreds of times. So, more dramatically is how this cost of human genome sequence decrease. So, as you may know, that the Human Genome Project, which is from 1990 to 2003, and cost like more than $2 billion. But till now, till 2004, the Illumina just announced that they have decreased the cost to like $1,000 to sequence the whole genome of a human. And I believe maybe in the near future this personal genome or human genome can be sequenced with less than $100, maybe perhaps hours. If this comes true, then we can essentially very easily do the genetic screening for much more sorts of cancer predisposing genes, or just do the cancer genetic screening or genomic screening. So that's how this DNA sequencing technologies provide us. So we can now directly screen sorts of cancer disposing genes with affordable cost. So for Angelina's case, it takes like $4,000, but maybe this year it will cost even less because you have higher sequencing capacity and you can pull more samples together. You can lower the cost. And with this technology, you can actually construct the catalog of cancer-causing genes for each major types of cancer. And also, you can profile not only the DNA mutation, you can also profile that the gene expression that linked with cancer initiation and progression, and also the epigenetic changes. And with all this information, we can develop more effectively to detect tumor in any stage and very accurately classify cancer types. As you know that for many cancer, it cannot be very accurately classified by this pathology. But now it's more and more accurately classified by genomics profiling. And we can use this knowledge to devise targeted therapy. In case you know where what causes this cancer or what mutations are involved in particular cancer, and also you can monitor the drug response. And in the end, I believe that maybe in 10 to 20 years, maybe we can make the personalized medicine in cancer prevention and treatment come true. This is some very excited development, one of the very exciting development in cancer research field and also in clinic. So let me just show you. Now there are several large consortium. They are trying to catalog the cancer critical genes. From the very beginning, you already know that different types of cancer are, in general, you can consider that as very distinct disease because they contain different mutations. The initiation and progression may be very different. So the idea is that by this sequencing technology, we would like to profile the mutation of each major types of tumor, and we take like hundreds of tumor normal pairs of samples. We need to know what are the mutations involved in each type of cancer, including the gene expression and epigenetic changes. This is what this International Cancer Genome Consortium are doing or has done. And then there is American sequencing consortium that is called the Cancer Genome Atlas, and the UK, United Kingdom, the Cancer Genome Project, they are all doing the similar things. Now with this, you can know that for these major types of cancer, how many mutations in each tumor, and then the next question is how many of them are really the cancer-causing mutations, and how many of them, which of them are drug targetable. Again, I will give you an example for breast cancer. Here shows the result that by this cancer genetic study, what are the mutations, the cancer gene mutations involved in what types of breast cancer. For example, the TP53, the PIK3CA, and so on and so forth. So you can accurately profile which genes are mutated and what is the frequency. And then you can classify this breast tumor more precisely, and then you can use this information, you can develop the targeted therapy or develop a drug more efficiently. So this is the third part, is about how we can use the information we obtain from cancer genetic study to really prevent and detect cancer in earlier stages. And I have emphasized that the revolution of DNA sequencing technologies greatly impute the cancer genetic testing and also targeted therapy. And I think this will eventually lead to personalized medicine in cancer prevention, diagnosis, and treatment. Maybe in the near future, in 10 to 20 years, the cancer research or cancer clinic will change dramatically. So this is the main content of this lecture. So in this lecture, I would like you to have a clear picture of what is cancer. Cancer is the tumor cells that is get malignant, and it's a very complex disease. And then what causes cancer? And I clearly showed that all these external causes and intrinsic causes have genetic links with cancers. And I introduced 3 types of major cancer critical genes, which are proto-oncogenes, oncogenes, tumor suppressor genes, and DNA repair genes. And then I discussed with you how these 3 types of cancer genes cooperate to trigger the cancer initiation and progression. And in the end, I have introduced how we can utilize this knowledge that we learn to study, to prevent and diagnose cancer, which is far more effective than cancer treatment. I would hope that by the end of this lecture, you have a much clearer picture of these 3 key questions of the cancer research field and also maybe for clinic. So with this, I will stop here, and I will thank you for your attention.